Welcome to SCigest, your fortnightly science from the Bioeconomy Science Institute.
Tēnā koutou katoa, ko Ki-Taurangi tōku ingoa. My name is Ki-Taurangi. I am a scientist in the Te Ao Māori team here at the BSI, the Bioeconomy Science Institute. Today I'm talking with Dr Toni Withers about the Endeavour program Protecting Aotearoa. But before I kick into that, Toni, do you want to give us a little bit of a rundown on your background, who you are, where you come from, what interests you about science?
Hi, Ki-Taurangi. Absolutely. I am a senior entomologist here at the BSI, and I kind of accidentally became an entomologist. I did a degree in zoology at Massey University. I'm a New Zealander. And then I undertook some postgraduate research on an insect pest, and that's how I became a passionate entomologist and have ended up doing that for my career ever since.
So were you interested in insects as a child?
Not particularly. I was much more interested in birds and trees and the environment, but once I learned about insects and how insects eat the plants, or a lot of insects eat the plants that birds and other animals also then interact with, I understood what an important role they play in the environment, and just because they're small doesn't mean they're not playing an important role.
Mm-hmm. And so have you always been more interested in pests rather than, say, endemic species?
Oh, I'm interested in both, actually, but it's really important to have some entomologists who work on insect pests, because they're a pest because they're eating something or threatening something that we value in New Zealand. So all our food that we produce has got some kind of interaction with an insect. Mm-hmm. And so if they're going to damage that food that we then value, or our crops, then they're important to us, to our society.
Yeah. And have you ever found a pest insect that you've secretly taken a liking to because it's cute or, it's a bit quirky?
That's a good question. I've really enjoyed working on the tortoise beetles, the eucalyptus tortoise beetles that feed on the eucalyptus trees that we value here. They're super cute.
They're so cute.
They basically look like a little miniature tortoise. And they've got wee pads on their feet that help them to suck onto the leaf surface so they don't fall off the tree when the wind blows. And they're quite likeable. Yeah, I
did a project in uni on that species, and I just remember looking at the pictures and being like, oh, such a shame that they're not great for our environment. Yeah. So I guess kicking into it, can you give us a bit of an overview of the Protecting Aotearoa research program? What is it? What's its purpose? What problems is it trying to solve?
Absolutely. The Protecting Aotearoa program is a 5-year research program that's been funded primarily by MB. We started in October 2023, and we've got funding until the end of 2028. And it's really a research program to understand more about how the winds bring pests and pathogens from Australia to Aotearoa's shores, and to try to understand that better, when it happens, and whether we can then understand more about how that mechanism brings those pests or those pathogens. Perhaps we can get to the stage where we can actually predict the times that are most dangerous, that have got the greatest risk for something new arriving here. It's all about understanding. The more we understand it, the more we're going to be able to predict it, and if we can predict it, then perhaps there's something we can do about it.
Okay, yeah. And so why is this research important for Aotearoa's biosecurity system right now?
Well, at the moment, our biosecurity system focuses a lot on the border, and that's because we already know that a lot of pests and pathogens get carried here on commodities through the channels that we all think about, so aeroplanes and ships, either hitchhikers or being hidden within things that either come by accident or are smuggled here. And so a lot of focus goes on that. Whereas the wind blows every day. We don't really think about that, the role that it might play in bringing something new. And so we do actually know that it does bring new things. So that's a slightly different focus of this research compared to most of the biosecurity research that we do.
Okay, well, you've already just talked a little bit about what I was about to ask you,
Oh,
but no, that's okay. What makes this program different from traditional biosecurity approaches?
We are really interested in the meteorology and modelling atmospheric movement. And for this, we're partnering with Earth Sciences New Zealand, previously known as NIWA, and the University of Canterbury. And we're really trying to understand the patterns of the wind, how it might be picking up pests and pathogens under different kind of weather systems in Australia, and then how they get transported across the Tasman. And the Tasman is about 2,000 kilometres, so it's quite some distance. And then we're also seeking to understand how those things might then be deposited from up high in the airstream down to the land, where they might then interact with the plants and animals that we value. So that's really quite a different approach than your normal biosecurity research.
Mm-hmm. I'd have to agree with you there. So we've known for a long time that pests can arrive here by wind. What has held us back from managing this pathway effectively?
When things arrive on the wind, we've tended to feel like there's nothing that we can do about that. For instance, when birds have arrived via the wind, our legislation suggests that they got here under their own steam, therefore they have now become a new native organism, such as when the spur-winged plover was blown to New Zealand and a pair was first seen in Invercargill, and there was much debate about what could be done about that pair, and it was decided that it had got here under its own steam, therefore there was nothing we could do about it. No one could shoot them, and so they've since spread all throughout New Zealand, and there's probably plenty of people that at some point would have wished that they hadn't made that journey. So we need to have a bit of a change in the way we think about these wind arrivals, because the wind arrivals can be a real problem for New Zealand, and one of the examples is the myrtle rust that we know arrived via the wind, and now that it's here, it's actually threatening some of our valued native Myrtaceae, and it is still considered an unwanted organism in New Zealand. And so there is something that we can do about these organisms if we find them soon enough. So if we know they're coming and we know that they're going to have a threat and have a negative impact, if we can predict exactly when they're most likely to arrive or what portion of New Zealand, then there is something we can do about it. Our primary industries that might be impacted by that pathogen or that new insect could actually go out and undertake surveillance, and the more quickly that you locate something new, the more likely you are to be able to successfully eradicate it if that's your aim.
Mm-hmm. It's interesting you say that about, birds becoming native. I heard a really interesting kōrero the other day that might need to be fact-checked, but I heard a kōrero, and maybe you know about it, about the swallows in Aotearoa and how they're considered to be native. And apparently they had come from Australia, right? But they had come on a ship from Australia, but nobody put them on that ship. They found their own way onto that ship, and because that ship came to Aotearoa, it was considered accidental or by nature. But then there was another interesting kōrero that they had originally come from another country anyway. They'd come from somewhere up in Indonesia and then travelled down through Australia, and then found themselves on the ship and then found themselves in Aotearoa and are now considered native.
Yeah.
So, I think that's kind of a very interesting way to look at it.
Yeah. And actually a number of our pests have first established in Australia before then making the journey from Australia to New Zealand. So Australia is one of the main countries that we need to be looking to and finding out what's impacting plant production over there in order to inform the risks of things arriving here as well.
Mm-hmm. So your team is developing a model that predicts when winds might bring pest moths or pathogens to Aotearoa. How does a model like this work?
The model is under development at the moment, and will rely on inputs from a number of places. So there'll be inputs in Australia or the Pacific Islands according to what the ecology is of particular pests and pathogens in order to inform the likelihood that they might get picked up by the wind. We then will have the direction of travel across the sea on the wind pathway will be directed by wind and meteorological data, which will be inputted on the time series during which any potential journey might take place. And then we're going to layer that with probabilities of survival. So depending on what the pest or the pathogen is, it's going to be subjected to forces that will limit its survival on the journey, because the higher up it is in the atmosphere, the colder it is, the higher the UV levels, the lower the oxygen. And so all those factors, including buffeting winds and potentially rain, all of those weather factors that they might experience on that journey are going to impact on their survival, and should they get deposited in New Zealand, it'll also have an impact on their likelihood of being alive when they reach the surface. So our model is going to be a probabilistic model that will incorporate likelihoods at each of different steps along that pathway.
Okay, well, I guess speaking of environmental conditions and survival, what kinds of environmental conditions are you looking at to understand whether the pest can start and survive the journey on the wind? So, what wind conditions are we looking for?
The factors we're looking at include temperature and air pressure and UV levels. We're going to try and look at the impact of rainfall as well. So bringing all those things together is going to be quite scientifically challenging.
And are you looking for, what wind patterns, is it if you're getting easterlies, northerlies, what are we looking for there?
Mm-hmm. We're looking at predominantly its wind direction, but not entirely. I couldn't really speak for the meteorologists because it's far more complicated than I can explain. But consistent westerlies, often the weather patterns that bring moths, for instance, from Australia, have been a cold front, so a cold frontal system sweeping over Australia, so southern Australia, New South Wales, and when the arriving cold front undercuts warmer air, that can lift moths up into the atmosphere, and then consistent westerlies associated with a revolving low will then carry those moths in ahead of that front across the Tasman to New Zealand. That's not the only mechanism, but we think that that's one of the common weather patterns that do enable them to arrive.
Okay. So if this model works in the way that you hope, what will it allow us to do in terms of early detection and response?
Well, in the future, it should allow us to predict if a certain season is looking like it's going to have a high risk of arrivals and which locations in New Zealand are going to be most at risk. So this will be based both on the historical data from other seasons that we have analysed, and then looking at the current climate and the patterns. We'll be able to inform MPI and the interested industries on where they should allocate their surveillance effort based on the risk profile of that season.
So that lets us get ahead of it and get ready to have that active response.
That's right.
Well, proactive, I should say.
Yes, proactive, yep.
So if we're getting into interceptions, you've been using interceptions of moths and butterflies to help validate these models. Why are these specimens so important?
These specimens are crucial. There's been naturalists catching tropical butterflies and moth species in New Zealand for many years, and there's been some papers in the literature that a lot of us are familiar with saying, the sightings of blue moon butterflies and other big tropical species have always caused interest to ecologists here. And of course we know that those butterflies can't breed in New Zealand, so therefore they must have been blown from, for instance, from Queensland or from northern New South Wales in Australia. So the sightings are very interesting, but the really useful thing for us is if we can intercept live specimens of large moths that we know are capable of making the journey and we can actually get a record of the time that they arrived. So the time and the day within a few hours is ideal, but at least within a day. And then each of those data points, if we can be assured that the moth has come from Australia, we can then download the detailed weather data, and we can analyse the possible path that those moths travelled and look at every hour on that trip what the conditions are that moth would have experienced. And we do what's called a back trajectory analysis. And then we're getting to the point of being able to say perhaps what part of Australia that moth came from.
Awesome. And so, what makes finding these moths and butterflies so difficult? Why is it like finding a needle in a haystack?
It sure is. It's like finding a moth in a haystack. It's very difficult. As you know, we've got a very long coastline. Our west coast stretches a very long way, and it only takes a very small shift in the wind to result in a moth landing either in Northland or the Waikato or Taranaki or even further south. And so we have been partnering with some end users and community groups in Taranaki. We decided that was a good place to focus on. But we can be out looking on the coast of Taranaki, and even that coastline is massive. And then we find that someone's spotted moths have actually arrived in the Waikato or in Auckland. And we've been waiting in Taranaki for them and not found any. And they've gone a little bit further north up the coast. So it's very challenging to be at the right place at the right time.
Okay, so going back to what you just said about, maybe volunteer citizen scientists, perhaps, you've had some surprising help from volunteer citizen scientists. Who are they? How did they become a part of the project?
Yeah, I'm happy to share that. It's been such a thrill to be able to reach out to people and get some volunteers to start looking for moths for us. The people need to be able to recognise an exotic moth from a New Zealand moth. And so that meant there's only, a smaller proportion of the population with those sorts of skills. And the right place to start was using iNaturalist, which is an online platform for people that are really interested or passionate about the environment. They can spot things in the environment, take a photo and upload that photo, and then it becomes a preserved data point. And through doing that, I met some more wonderful entomologists. Some of them are scientists, some of them are what we call citizen scientists. They're naturalists who have decided they love moths and love taking photos of them, and a lot of them just take a photo and then let the moth go again. But each identification from a good quality photograph becomes useful for us. And at first I started making a record of all the sightings of Australian moths and looking at the weather patterns on the days that they had arrived, and I found there were some good patterns, with more arriving shortly after or during westerly winds than, say, during consistent easterlies. And then I started reaching out to some of these people, and I've now got a network of 5 or 6 really keen naturalists spread down the North Island who often have a light trap at their own house where they attract moths and see who's arrived that day, and they take photos. And so I had to reach out to them and tell them about this Protecting Aotearoa research program, and tell them or ask them, would they be prepared to sacrifice the life of the occasional moth if they thought it had come from Australia. So that was quite a hard conversation, because the more you love creatures, the less you want to see that creature die. Mm-hmm. But this research program requires us to have the specimen. And so I reached out to a few of these naturalists, and a couple of them were very clearly said no, they wouldn't sacrifice the life of a moth for this lady who seemed to want the specimen. But there were others who understood the value of what we were trying to do and said, sure, they'll catch them for us and pop them in the freezer, and record the time and the date and what the weather was on the day they found them, and passed the specimen to me. And so it's been absolutely fantastic. Oh, you asked about who some of them were?
Mm-hmm.
One of the ladies is Tansy Bliss. She's a ranger with the Pukorokoro Miranda Shorebirds Trust on the Firth of Thames, and although it's not on the West Coast, she's been successfully capturing quite a few awesome specimens of Australian moths for me, and the Trust was really thrilled to be able to assist with this piece of science and support her doing that for us, so that's fantastic. And the other group that are doing an amazing job is we also reached out to a company called OMV New Zealand, and they are a company that's operating some of the largest oil fields in the Taranaki region.
Wow.
And they have a couple of offshore platforms. So you might have heard of the Maui field. So there's a platform out quite a long way off the Taranaki coast called Maui B. There's also a floating platform south of Taranaki called the FPSO Raroa. And we talked to the staff there about whether they had anyone on those platforms who would be interested in capturing moths that they might intercept, because these platforms have got very large lights on the platforms.
Of course.
Yeah, they don't want any ships or boats banging into them in the night, so they have these massive lights. And as you know, moths are attracted to lights at night. And we've known historically that some tropical butterflies and moths have been caught on these platforms in the past. So we reached out to the staff and they were willing to assist us, and they've got skeleton crew that have shifts working out on those platforms, so they're always manned, those two in particular. And so we've got some fantastic volunteers who run around the decks every morning and check to see whether any moths have arrived, and are sitting under the lights at dawn, and they grab them up for us and pop them in the freezer, and are supplying some really fantastic specimens and data to us.
That's amazing. I really believe that citizen science is underrated, big time. Underrated, underutilised, and kind of going back to, you know, it being a little bit difficult to maybe find people who can identify these moths. Do you think that there is, might be a silly question, but do you think there's a big difference in how New Zealand native moths look in comparison to Australian moths? You know, are they very physically different?
Well, some of them are. Some of them are really unique here, like our purere moth. Mm-hmm. You can't really compare that to any other, so that's very unique. But a lot of our other large moths are actually the same as species that are in Australia. So we can suppose or hypothesise that a lot of those large moths actually flew from Australia to New Zealand at some point after Gondwana land separated. So whether it was however many eons ago, we will never know. But a lot of them do look the same. They also have cousins, so species in the same genus that look very, very similar. So it might just be, so the species we have here might have a little kidney-shaped marking on their wing, but there's a species that's not here yet that there's a little black, like a little wee arrowhead at the top of that kidney-shaped marking. And so if you know what you're looking for, you can say, oh, you know, ahoy, I've found an Australian. But if you don't know what you're looking for, you would have no idea.
Yep, so that's why it's so difficult, because the differences can be so subtle.
Yeah.
Okay, all right. So going back to talking about OMV, why are offshore platforms like OMV installations such valuable observation points for wind-dispersed species?
That's a good question. I think although when we get a moth on an offshore platform, we can't say for certain that it would have been able to make it to the coast. Perhaps it was just running out of energy and it was just starting to fly lower and it saw the light and just dropped down to the light, and it wasn't going to make it. But chances are,
if it got that far,
if it got 1,940 kilometres, it was going to be able to make the final 55k to the coastline. But the great thing about it is that it gives us a really good date and time stamp of the time of arrival, within a few hours anyway. Perhaps the crew weren't out at midnight, but if they were out at 7 o'clock in the morning, that means that they probably only arrived a few hours before, and that can then provide us with really detailed data on which to look back at the wind, the vectors of where the wind was moving on that night and the day before.
Okay. So, even if it's just from a funding perspective, how has the involvement of naturalists and volunteers changed what's possible for this research?
It's really lifted the game, because we were only going to be aiming to get data from Taranaki, whereas now we've got some really fantastic data points of moths that have reached other parts of New Zealand. And so Auckland and, for instance, the Firth of Thames moths, we're now, because when we work with our stakeholders who are interested in the outcome of this research, a lot of their questions are, if we are growers in a different region, how's your model going to help us? And so now I can at least create the data from some different regions of New Zealand and give the modelers an opportunity to look and see whether the pathway might have been different for a moth that arrived in Northland versus one that arrived in Auckland or Taranaki. So we don't know yet what the bounds of the error, basically we don't know how wide the error bar will be yet on how detailed our model will end up being, whether it can predict that one region is going to be more at risk on a certain day than another region, but at least we've got the inputs to give the modelers the opportunity to look at that.
Okay, so and earlier you mentioned us getting information on where the moths come from. I'm imagining there might be some very specific techniques you use to figure that out. Do you mean sort of which country, or do you mean which region, or how do you find that information out?
Yeah, well, it's pretty neat actually. So what we're going to do with the moths that we've managed to get their specimen, so we've got their body safely in the freezer, what we can do is we can actually look at the traces of their body composition. And when you eat a plant, let's just say, when you're a caterpillar and you're eating a plant from a certain place, that plant contains certain minerals and biogeography traces from the soil that it was growing in. And so when you eat the plant that was growing in the soil from a certain place, you can actually incorporate a little bit of that same
signature?
chemical. Yeah, same chemical signature into your own body. And the moths do exactly that. In the chitin that makes up their exoskeleton, there will be traces of where they were eating when they were a caterpillar. And so by undertaking analyses on their bodies through mass spectroscopy, we can find out whether they came from Australia or from New Zealand and perhaps from New Caledonia. So we should be able to separate them by country, and depending on how well our data set goes, we might be able to even separate them into region. So we might be able to say, that Australian moth is showing a signature of chemicals from the Queensland border with New South Wales rather than from Victoria or South Australia. So, at the very in another year or so, we'll have the data on each of our individual moth specimens.
Okay, and so what does that add to your ability to test and refine the wind dispersal model?
It's going to be really important. So the modelers are going to be waiting for the data. So they're desperately waiting for the data so that they can do the back trajectory analyses. So we can say this moth was caught in Taranaki, and they're like, but what part did it come from? And when I can finally say, oh, it arrived on this day at this time, and it's got a body signature suggesting it was from South Australia, then they can refine their back trajectory modelling.
Awesome. So I guess looking ahead, what excites you the most about where this research is heading?
Oh, all of it. It's all exciting. And the neat thing with the group that I'm working with is that we're really quite diverse. So our program leader, Dr Ilse Pretorius, she's gathered a really varied team together. And so we're working with meteorologists, we're working with modellers, we're working with statisticians, with ecologists, with a whole range of scientists, and yet we're all working together towards this common goal, which is to get this model that can take all these different and varied things and pull them all together to create something useful. So yeah, it's really neat working with all those different sorts of minds, and we're all very specialists in our own field, but Ilse is able to have an eye on where we're heading, which is this one output.
Yeah, she's awesome to work with. She's an amazing early career researcher, and she's so giving and open with the information and knowledge that she holds. So I imagine she's really cool to work with for you as well, to work so closely with her.
Yeah, she's a real inspiration.
So you mentioned working with lots and lots of people. We've got our volunteer citizen scientists, which is awesome. How often do staff from the BSI, the Bioeconomy Science Institute, head over to Taranaki to do light trapping and stuff like that? And how do you predict when they should go?
Well, we've now got our meteorologist from the University of Canterbury, Marwan. He is keeping an eye twice a week on the weather patterns, and he can run a model that can help to inform him of whether the weather patterns look similar to ones in the past that have resulted in consistent westerlies. And then he'll drop us an email and go, low likelihood of an air bridge. This is what we call them, these connecting westerly winds. We call them an air bridge if it looks like they're coming from Australia to New Zealand. And so he'll do a shout out and say, it's looking likely, guys, get ready. And just when those air bridges are forecast, and when we've got a team that have got the time to go, then we gather around and head over there. So yeah, it's probably working out at about once a month or so, but air bridges are more likely in the spring and the autumn than any other time, we think. So we're on alert during those times of the year.
Cool. So the work that you've been doing and the rest of the group have been doing is quite novel. How might it change the way that Aotearoa manages future biosecurity risks?
It's probably too soon to say how it will change the management of biosecurity. The biosecurity research is a massive spectrum. It involves a lot of government departments, end user groups. It's quite a complex beast with a different group of stakeholders looking after each particular, what we call pathway, and trying to minimise the risks of each individual pathway. And shout out to MPI, they do an amazing job. So it's a little bit too soon to say who will manage or keep an eye on the aerial pathway, but we think by the end of our program we'll get quite a good feel for whether that's going to be a primary industry group or whether we're going to have some freely available software, or whether it needs to sit with earth sciences or something like that. Don't worry, we'll make sure that everybody knows at the end of the program what the next steps are going to be.
Awesome. So slightly off topic, but still sort of on topic, we have the BSI. So we've recently merged from plant and food research, ag research, Scion and Manaki Whenua Land Care research to become the BSI. What do you think, or how do you think this might impact the program Protecting Aotearoa?
Yeah, thanks for that question. I can see it assisting us. So as a scientist who was previously in one of those Crown Research Institutes, Scion, it was much more difficult to work with colleagues in the other Crown Research Institutes. You had to formalise, a lot of your collaborations had to be formalised into contracts and costed, and money had to change hands, and you couldn't start work until all the legal teams had agreed, and so there was a lot of effort wasted in paperwork. Whereas now it's going to be much smoother and simpler to work together with other colleagues that have got either a common interest or have got a different skill set and you really need them. You're going to be able to just ring them up and go, hey, have you got time? Would you like to do this? Would you like to do that? And it's just going to smooth the pathways for really useful connections between scientists from different disciplines.
Yeah, definitely having more money for the actual science rather than spending it on admin. I think it's a really positive change as well.
Yeah, well, we can hope so, and I'm sure that's the direction that it's going to head.
So if listeners want to support this mahi or learn more, what is the best way for them to get involved?
Well, they can, if they're really interested in this particular program, they can sign up to our newsletter. We have a newsletter called Blown Away, and you only have to click on the link which says sign up to our e-news, and every 6 months we can send you a wee newsletter letting you know how we're getting on. But the other thing that I always like to say to people is every person matters. All of us, we're the eyes of Aotearoa, and we have a part to play in biosecurity because it'll be one of us that will spot something new first, and you never know if you're the first person to capture something, at least take a photo of it. If you can put it on iNaturalist, which is not difficult these days with smartphones, if you can learn to take a photo and then select a photo with a little tick on that photo, and you have access to the internet, then you can either through the app or through a website page, you can upload a photo, and then this amazing iNaturalist tool and the experts who watch the pages will identify that organism for you. And you might be the one who finds something new and starts an important piece of work in either eradicating something nasty or finding something new that's not nasty. I mean, there's still new indigenous endemic insects being discovered in New Zealand. There's still being new species are being found, and you might have a bug named after you. But it's not going to happen unless you go out there and you make the effort and you look and you take photos and you just play a part.
Okay, so before I ask the last question, are there any other topics or anything else that you want to talk about that you feel we may have missed?
No, I think I hope this has been of interest to some of the listeners, and I know it's a neat program to be involved with, so I hope that there was someone who was interested in what we had to say.
I can guarantee you that there will be. Okay, then last question. What is your favourite insect? Right now. I mean, I know as an entomologist it must be a very hard question. So what comes to mind? What's your favourite insect right now?
I think the one that jumped to mind was the giraffe weevil. They're super cute.
Guess what? It's my favourite too.
Oh, there you go. There you go. Yeah, they're just awesome, and I've got some in my, well, not in my garden as such, but right on my back fence. I have a bush reserve, and I've seen them come out of the little holes in the trees there. So it's pretty neat. They're my neighbours. So
yeah, I've got this massive Coleoptera book, and it's got all these pictures, and they're all life-sized and whatnot. And the giraffe weevil's in there. It's an international one, and the giraffe weevil's in there. And when I saw it, I was so happy.
Yeah. No, yeah. that long rostrum, that long nose that they're known for. Yeah, it's pretty hard to get something cuter than that.
I think scientifically it's called a snoot. Okay, well, thank you so much, Toni, for joining us today. Thank you for your mahi, thank you for your time, and we look forward to seeing the future of protecting Aotearoa.
Thank you very much. You're most welcome.
And thank you out there for listening. Please check out our SciGest pages for extra links and photos on the topics that we've covered today. Please subscribe to our channel on iTunes or your favourite podcast provider, and don't forget to post a review and rate this podcast.